All right, the end of chapter 2 really goes over the 4 different macromolecules. These are going to be carbon-based molecules that are important in the body. We talk about them a lot in nutrition, but they're part of all the different structures in the body. So real quick, before we jump in here, let's make sure we're good on this idea of organic versus inorganic molecules. So for example, we've spent a bit of time already in this chapter talking about water. That's a very important inorganic molecule. Or sodium chloride, table salt, that's an important inorganic molecule. When we start looking at organic molecules, you got it, they are carbon-based, right? So they're going to include carbon, and technically they need to have carbon and hydrogen. Right? So usually we talk about organic molecules as if they contain carbon, but one of the important inorganic molecules that we'll discuss contains carbon, right? CO2, carbon dioxide, is inorganic because it doesn't have hydrogen. When we look at some of our organic molecules, you might expect to see something like glucose, C6H12O6. Right? It has carbon and hydrogen. Now, a reminder, when you have those subscripts, right, they're telling you you're dealing with a molecule. We know that these elements are covalently bonded. And recall that this tells us there are 6 carbons, 12 hydrogens, and 6 oxygens in this molecule. Okay, so let's go ahead and actually look at the 4 macromolecules that you'll need to know here in anatomy. Do you remember these from Bio 1111? We just mentioned one, which is a carbohydrate. Most people do better if they start thinking about foods. So carbohydrates, lipids, proteins, and then the one that's not really food are the nucleic acids. Right, and that's your DNA and RNA. So let's jump in here. So this is a table that's kind of general about organic compounds, and you would have seen something similar in Bio 1111, but it was actually a much longer list. So your textbook is trying to narrow this down. They're basically using R here as a placeholder. It could be anything. And they're talking about what they're calling functional groups. This group that's kind of hanging off that R, that base, that, not base like basic, but that core part of the molecule. Don't worry about these too much. We will talk about this idea of an amino acid. One end is an amine group, which acts as a base because nitrogen can actually accept a 3rd hydrogen. This carboxyl group, where you have a carbon double bonded to an oxygen, and that carbon is also bound to an OH. This often acts as an acid because that hydrogen can come off. So we'll see both of those functional groups in an amino acid. The hydroxyl group, that OH, we talked about that a little bit with pH. So you'll see that come up as well. That becomes important if you're trying to link molecules together, and we talked about a dehydration synthesis where you take the OH off one and an H off another, right? That might be a way to link these molecules. And then this phosphate group, you will see a lot. Even if you just think about something like ATP, right? So this core group here might be the adenosine and the first 2 phosphates, and now they're showing you this phosphate group. So I wouldn't sit there and try to memorize these names. They're going to come up here as we talk about these different macromolecules. All right, so let's start off with carbohydrates. I'll find a better marker here. Right, so carbohydrates. Basically, all carbohydrates are going to fit this ratio, right? 1 carbon to 2 hydrogens to 1 oxygen. So for example, here they're showing us glucose, which I had just written out, C6H12 O6. So see how the hydrogen is doubled what the carbon and the oxygen are. It is literally a hydrated, right, water added to a carbon, carbohydrate. That's where it gets its name. Now, maybe this is a great place to talk about this idea that when we look at organic molecules and they're carbon-based, it's like, well, why carbon? So carbon, if you look at your periodic table, carbon's atomic number is 6, right? It has 6 protons, 6 electrons. Take a moment, hit pause, and try to arrange the electrons in carbon, and then start back up and see if you did it the same as me. Right? Electron, electron. 6 electrons. There are 4 electrons in this outer shell. Now, this puts carbon in an interesting position. It wants 4 more, but losing 4 would actually be kind of extreme, right, to get down just to that inner shell. But gaining 4 requires some work. But what happens is carbon then, right, you'll notice seeing it have 4 bonds, that lets it make these long chains. It lets it form rings. And so the carbon, because it has 4 valence electrons, can form much more complicated molecules than a lot of the other elements that we were looking at, right? And so that's really why life is based on carbon. While we're here, let me point out one more thing. So this is a picture of glucose, and you'll see this a lot. Chemists, to do this in a shorthand, right, you might see me just draw a little ring structure like that. Technically, anywhere there's nothing written is a carbon. So I won't because it's too much work, I should draw glucose like this, right? But this is a carbon, 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 carbon. It's just, and carbon. It's a shorthand. So if you see that, that's what's going on. Okay. All right, so carbohydrates, a hydrated carbon. Now, carbohydrates are a really good source of energy. They have about 4 kilocalories per gram of energy, and this is energy that your body likes to use. Muscle likes to use this, brain really likes to use this. And carbohydrates come in a few different forms. In fact, as we look at all of these macromolecules, we'll see a simple form, what we call a monomer, a single of something, and then a more complex, maybe a chain or something like that, that we talk often, like in this case, we'll talk about polysaccharides. So the simplest form of a sugar is a monosaccharide. Right, a single sugar. And you actually have 3 different monosaccharides: glucose, fructose, and galactose, which they're not showing up here, and that's fine, right? But each of these rings is a monosaccharide. So what they're showing you here is the process by which we can put 2 monosaccharides together and form a disaccharide. And a disaccharide is going to be 2 sugars put together. Notice the process that takes place in this reaction. It's a dehydration synthesis. So we're actually removing the hydroxyl group off of one of our sugars and a hydrogen off the other. And by pulling that water out of the molecule, you're able to form a bond and hold them together. See, we said there'd be all sorts of water working around. The reverse then is also true. If you take sucrose, which is a glucose and a fructose hooked together, and you insert water, you can run a hydrolysis reaction, right? Remember, lysis was splitting with water. As you force that water molecule in, you can pop that disaccharide apart into its individual monosaccharides. The disaccharides that are important in the human body, sucrose is probably the one that you hear about most. That's going to be your table sugar, right? And that is a glucose and a fructose put together. Lactose, sorry. is going to be a glucose and a galactose, right? And that is milk sugar. So if someone is lactose intolerant, what's actually happening is their body is unable to run hydrolysis and split this disaccharide into its components parts. Remember, each of these arrows showing the reaction taking place, there's an enzyme behind that arrow helping the reaction occur. Someone who's lactose intolerant typically is lacking that enzyme and therefore can't digest the milk sugar. The last one is maltose, and that's actually just two glucose molecules hooked together. Okay, then of course we can actually make much longer chains and we can form some polysaccharides. Right? So poly is going to indicate much longer collections of sugars. In this case, they're showing us glycogen, which is a very important carbohydrate in the body. This is how you're going to store glucose for later. So both the liver and the skeletal muscles are capable of hooking glucose molecules together into these long chains and then popping them apart when you need that energy to exercise, because you slept through the night without eating and you haven't had breakfast yet and your brain wants glucose, those sorts of things. So it's this kind of complex molecule, it's all branchy. Other polysaccharides that you're gonna see are going to be things like starch. And this is how plants store glucose in long chains, but it's not branched, it's just a straight chain. And you can break that apart and get energy from it. And plants also, cellulose, make long chains of structural carbohydrates called cellulose, and you're actually unable to break those individual sugars apart. You don't get any energy from cellulose. In fact, in our diet, we just call it fiber. It's going to go through the body without providing energy. But it is important, right? That fiber is roughage, very important in digestion. So again, pointing out monosaccharides like glucose and fructose, the disaccharides that we listed, polysaccharides, they're talking about glycogen. That's the only storage form that humans have, but of course, starch and cellulose are important in the body. Okay. Look at this, you're getting like half your nutrition class in this chapter as well. Okay, so let's look then at lipids. So again, we see lipids being carbon-based, lots of hydrogen in here. We know it's an organic molecule. Lipids are a great way to store energy, and the human body can use that as energy as well. The reason I say it's great is you actually get 9 kilocalories per gram of energy out of a lipid. So you can store twice as much energy in half of the weight. And while a lot of us tend to think of lipids as a bad thing, they're really important as a fuel source, they're important as insulation in the body, they're important for cushioning, protecting vital organs. So lipids are not a bad thing. We just need them, like everything, in moderation. So they're just showing you here a couple of examples of how we build these long chains. And a couple things that'll come up, saturated versus unsaturated fatty acids. If you recall from 1111, that saturated fatty acid, every carbon has 2 hydrogens attached to it. It's full of hydrogen, it's saturated. An unsaturated fatty acid will actually get rid of a couple of those hydrogens, and to make sure each carbon still has 4 bonds, they double bond, right? They share 2 pair of electrons in that covalent bond. And now we don't have as many hydrogens, we're not as full, so we're unsaturated. Now, what really matters here is that saturated fatty acids, if you put them in a form, like the way that we store lipids are as triglycerides, and a triglyceride is going to have a 3-carbon molecule called glycerol, and then 3 of these fatty acid chains. Oh, I'm not good at drawing this. Now, if those fatty acid chains are saturated, notice this repeating pattern. These are able to pack tightly together. Saturated fatty acids pack tightly together. Man, I am still looking for the perfect marker here. These pack tightly together, right, and are able to clog arteries. Clog arteries. Unsaturated fatty acids, when you throw this bend in the molecule, you don't hit that same repeating pattern, right? And now the tails are going to be looser. These are going to be liquid at room temperature, and then also in your blood vessels, so they don't clog them up. So this is like olive oil, where when these pack tightly together, they're going to be a solid at room temperature, like butter, right, and clogging up arteries. Anyway, so in your lipids, fatty acids get packed together as triglycerides, and that's our storage form. That's what you would find in adipocytes. Okay. It is again dehydration and hydrolysis that actually builds these molecules. So they're trying to show you here that basically you can you can form triglycerides out of fatty acids by pulling water molecules out, by doing a dehydration synthesis. That is anabolic. It's building something much more complex than these individual parts. But to break it apart, you can put water back in. You can run a hydrolysis reaction. Okay, other groups of important lipids. These are all what you might have heard of grouped together as steroids, right? Cholesterol is actually the parent molecule of these steroids, right? Meaning that if you want to make estrogen or testosterone, look at how similar these actually look, right? A few things added, taken off. That you need cholesterol, right? So cholesterol also is not bad. You need it to make these important hormones, right? So this lipid has a very different structure, kind of a ring structure based off of cholesterol, right? So we've got triglycerides, we've got steroids. Let's see. There's another group, these are the icosanoids. I know that's not what that, like that's a weird word. Icosanoids. These will come up a couple times. They tend to be chemical messengers. Prostaglandins are the one that you're most likely to have heard of if you need an example there. And then probably the last group that I don't have a picture of, the last group that'll come up with us are the phospholipids. Which are important, of course, in cell membranes, right? Remember that phospholipid bilayer? And it's actually interesting, this isn't that different than something like a triglyceride, right? In fact, you use that same 3-carbon glycerol molecule, and oh boy, that's a fatty acid tail, that's a fatty acid tail, and then you put a phosphate group up here as the head, right? And that's a phospholipid. Okay, let's look at our proteins. Proteins are so important. Oh my goodness, this is driving me crazy. Let's try one more. I will have to remember to bring fresh markers to this class. Here we go. Proteins, so important in the body, right? Typically we think of proteins as part of something like skeletal muscle being important in movement, but proteins are going to make up antibodies as part of your immune system. Proteins make up most of your skin that's protecting you. Proteins are going to make up some of our hormones. We use them in the bloodstream as buffers to prevent big swings in things like pH. They get used as enzymes, right? We mentioned that enzymes are made out of proteins. in cell membranes where they're embedded. They're really important in transport across the membrane. So proteins are really important. And if you think about it, your DNA, the code that you're carrying around, is how to build all of these super important proteins. Anyway, proteins are made up, the monomer, the simplest form here, is going to be an amino acid. Okay, and so all amino acids have this same structure. There are 20 different amino acids that we use in the human body. They all have a central carbon. On one end, they have an amine group. This has nitrogen and either 2 or 3 hydrogens, right? So it's able to accept hydrogen. It can act as a base. This end, that carboxyl group, remember, is able to act as an acid by donating hydrogen. So amino acid, right? Sorry, and this R group is just a variable. So there's 20 different structures that could be on that side of that central carbon. Okay, so basically those amino acids are going to get put together in long chains. So we go from amino acids to polypeptides, right? A long chain of those amino acids, and then that polypeptide is going to get folded into our actual protein structure. Okay? Once again, the way that we actually put amino acids together is through a dehydration synthesis. We see that water molecule getting pulled out to the side here and a bond forming, right? If you want to break that bond, you use hydrolysis and you can split those individual amino acids apart. Okay, this is what that folding process looks like. If you recall this from 1111, you have this polypeptide chain that is considered the primary structure. The order of polypeptides is dictated by your genetic code. Then the interactions between those different amino acids are going to allow it to fold into what we call a secondary structure, either as a helix, they call that the alpha helix, or it folds more like sheets of paper, what they call the beta pleated sheet, right? That's the secondary structure. And it's really not until the tertiary structure, right, where all the helices and folds are coming together that we actually get the functional protein, right? That tertiary structure is actually the really important one where a protein takes on its function, whether that's as a buffer or an enzyme or a structural protein in muscle, whatever. Now, some, not all, some proteins actually have a fourth or quaternary structure where multiple tertiary structures come together. Hemoglobin that we'll talk about in here, collagen are both great examples of that, right? But this is where we get our function. Now, there are things like big swings in pH or heat that can break that tertiary structure, right? Disrupt the folding, and we call that denaturation, or to denature a protein. So to denature a protein is to cause it to lose that tertiary structure, right? To break it down in a way where it no longer functions. And again, things like swings in pH or high levels of heat cause that. Just like frying an egg, right? That is albumin, the white of an egg is albumin, that's all protein. And when you heat it, it goes from clear to white. You're actually changing, you're denaturing the protein there. Okay, so again, when you think about things like enzymes, we looked at this picture in the last video. If you denature an enzyme, that site, that active site is going to change shape and that enzyme is no longer going to function. In fact, enzymes get used to catalyze all these reactions in the body because you can catalyze reactions using things like pH and heat, right, like frying the egg, but that destroys the tissues and things around it. And so we, instead of using those things, use proteins. Okay, last macromolecule are the nucleic acids. So nucleic acids, DNA, RNA, and the kind of oddball out is ATP. People always forget this one, okay? When we're looking at any of these molecules, they all have this same basic structure, right? So they have a sugar and a phosphate and then some kind of nitrogen-containing ring structure. Right? So in DNA and RNA, you probably remember your A's, G's, T's, and C's. Remember, uracil gets traded out. We use thiamine in DNA and uracil in RNA. Does that sound familiar? Remembering that? Anyway, for me, don't worry about memorizing who's a purine, who's a pyrimidine, right? But realize that these molecules are nucleic acids. Right? DNA then, of course, is in this famous double helix where we always have a pyrimidine binding with a purine across as kind of the rungs of this ladder. The big thing here, right, is remembering that DNA is the code to build proteins. It is the blueprint. RNA is a copy of one side of the DNA, right? And that RNA can go to the ribosome where the actual instructions are followed to build that protein. Okay. ATP, notice, sugar, nitrogenous base, and phosphate. So that is why it is still in this group that we call nucleic acids. ATP is important for what? Energy, right? It's the cellular energy. And the thing to keep in mind here, this will come up later in the year when we talk about metabolism, but it's really the bonds between the phosphate groups are where the energy is stored. In fact, if you look closer at these phosphate groups, they each have a negative charge. And so if you think about trying to put two negative poles on a magnet together, like they repel. So picture when you try to build ATP, you're pushing the magnets together. It's very difficult, right? But your body knows how to do that, and it's almost like loading a spring, right? Because now when one of those phosphates come off, it can release all the energy that's in that bond, right? Or think about setting a mousetrap. It's really hard to pull that bar over, but you're loading the energy. And man, when you break that bond, you can catch a mouse. Okay, so ATP is that cellular currency of energy. Okay, I'm gonna give you a little summary here in table 2.7, right? So we were just talking about our 4 different organic molecules. These high energy compounds, they are nucleic acids. I don't like how they separated that. But again, inorganic molecules, they can have carbon, right? But they don't have carbon and hydrogen. So as you go through this, again, this was a chapter or 2 in Bio 1111, so I know this is a really quick review, but you want to have enough kind of working ability with this language, have this, you know, somewhere fairly easily accessible in your brain so that when you hit some of these chapters, you're not having to dredge this up again. So that's why they give you this review early in anatomy, but hopefully as we use it throughout the semester in context, it'll start to make even more sense for you. All right, we'll see you in class.